Hello and welcome back to the another part of the video series. So we has been discussing about the structure of DNA, what is DNA, what is its structure, what it is made up of, as well as we have discussed about certain properties of the DNA in detail. In this part of the video, we are going to discuss a process which is known as replication, which majorly takes place in the DNA, and it is a very interesting process. But in this part of the video, this video series will be of three parts. First is the enzymes, what they are involved. Then second part will be regarding replication in prokaryotes, and the third and the final part will be the eukaryotes. So what are the enzymes which are necessary for the replication process, and what are all needed for the replication process, we will study in this video. So let's start with this one. So first of all, let us know what is replication is all about. So what is replication? Replication is the formation of identical DNA molecule. In this replication process, the DNA forms its own identical molecule. That's why they are called replica and the process is called replication. Isn't it interesting? Yes, it is. And this process takes place during the cell cycle. What is cell cycle? You might have known that the mitosis and the meiosis is a cell cycle process, and during the S phase, mainly in the interphase of the cell cycle, during the S phase, this DNA replication takes place, and finally they will be separated into the two daughter cells. They will be divided into the two daughter cells. Okay, so this is the property which is mainly takes place during the S phase of the interphase. This is to be important to be noted down. Wait one second. Okay, this is an important thing which is to be noted down. S phase of the interphase. It is mainly asked where does the replication takes place. So during the S phase of the interphase. And what does this process do? It forms the two identical DNA molecule. Okay, and the DNA replication is a semi-conservative. What is semi? Semi means half, conservative means to conserve. That is, it conserve one strand of the parental strand and another it will be form the new strand. That's why this process is known as the semi-conservative process. And how we came to know? An experiment was conducted. By that process we came to know that the DNA is a semi-conservative process. Okay, and there are certain enzymes as well as proteins which are involved in this process. First is the DNA polymerase, helicase enzyme, primase enzyme, single stranded binding protein, topoisomerase and ligase. These all 6 enzymes are involved in the process of replication. So one by one we will be going to discuss in detail what are they and what are the function of each of them. Let's start. Before that, this is the clear picture of how the replication occurs. When we will be discussing about the process of DNA replication, that time also I will mention how the process one by one what happens, they will discuss. For now, let's discuss first what happens. The helicase enzyme, you can see this is the helicase enzyme, this arrow mark. This helicase enzyme binds to the one strand of the DNA. Okay, and now what the role of the helicase enzyme is to? The helicase enzyme is to open the two strands, because if the replication is to be done, then the strands need to be open. Otherwise, how the replication will take place? So that's why what does the helicase enzyme do? Helicase enzyme binds to one strand of the, these are the two strands of the DNA. So it binds to the one strand of the DNA and starts opening it, as a result of which a replication fork will be formed. Then what happens? Immediately after opening the two strands, the single stranded binding protein, which you can see here, these are the single stranded binding protein, starts binding here. Okay, this single stranded binding protein will start binding to the strands. What is the in the both the strands? Why? What is the role of this single stranded binding protein? Because when the strands are open, now they are mainly at a risk of getting attacked by the nuclease enzyme. What is a nuclease enzyme? Nuclease are the enzyme. Nuclease means they cut the nucleic acid. Okay, so the nucleus, they are the risk of being attacked to the nuclease enzyme. Also, if the single stranded protein will not be attached to these strands, then again these two strands will join to each other, because these strands are complementary to each other. In order to make them separate and to hold them in this position, these two strands are attached by the single stranded binding protein. This is the role of single stranded binding protein. And what is the role of helicase enzyme? Is to open the two strand. Next, coming to the DNA primase. What does the DNA primase do? DNA primase is a short RNA form. It is a primer which provides a 3' OH site for the DNA polymerase to get attached, and the process of, that is, this is the DNA polymerase, you can see. So DNA polymerase, DNA primase will be attached, and RNA primer will also be attached at this end, and then what will happen? The elongation or the process of replication will start. So for the binding of the DNA polymerase, the RNA primer is present. And after that, what last enzyme which is responsible is the, next is the topoisomerase enzyme. What is topoisomerase enzyme? So while opening the strands, what happened? The other end of the strands gets superly coiled. So in order to get this coiled free, so that they can easily, smoothly, the replication can takes place, that's why the topoisomerase enzyme is responsible for regulating the supercoiling. Okay, so to release the tension which will be created, so this front part will get open and the back side will get superly coiled, so in order, so that the process of replication takes place smoothly, the topoisomerase enzyme is mainly relax the supercoiling. Okay, so the topoisomerase enzyme is also added. Next, coming to the ligase enzyme. What does the ligase enzyme do? Mainly, after the DNA fragments are formed, they seal these fragments. They seal the Okazaki. What is Okazaki fragment? What is lagging strand? What is leading strand? We will be all discussing while we will be discussing about the process of replication. But for now, it is necessary that what are the enzymes which are responsible in the replication process. So overall, I have told you the functions of each enzyme, but now each one we will read in the detail. First, we will deal with the DNA polymerase enzyme. So what is the role of DNA polymerase enzyme? Its main function is to replicate the genome. So you have seen in this diagram that once the DNA polymerase will be attached to the primer region, then what happens? The replication process will start. So it is responsible for replication and allow the genetic transmission from one generation to another, because by the process you have seen that in the cell cycle process the replication takes place and they will be divided into two daughter cells. So due to replication, as these DNA polymerase allow the replication process to happen, so the genetic transmission from one generation to next generation will be taking place. And main point is that the integrity of the genome, the constitution of the genome will be maintained. Okay, and the synthesis always takes place from the 5' to 3' direction. This is a very, very important point to be noted down. The DNA polymerase synthesis takes place from 5' to 3'. Then DNA polymerase is of two types, that is DNA dependent and another one is RNA dependent. DNA dependent is mainly depend upon the DNA template, okay, and RNA template is known as reverse transcriptase, which is responsible for formation of cDNA and repair process, which we don't need to be dealing in detail of the cDNA. What is cDNA? We will be discussing in later somehow of the video. And both of this DNA and the RNA dependent DNA polymerase requires primer. So they can't function without the primer. The primer may be DNA or RNA. Okay. Next, moving to the three types. Basically there are 5 types of DNA polymerase. The 4th and the 5th type is responsible for the error proof reading, and these 3 are main type of DNA polymerase which is found in case of prokaryote. And this DNA polymerase 1 have both the 3' to 5' and 5' to 3' exonuclease activity. You must be wondering what is exonuclease activity. Nuclease means to cut the nucleus, nucleotides. So mainly to cut the primers, this exonuclease activity is performed. As they cut in externally, they are known as exonuclease. If they cut internally nucleotides, if they cut the nucleotides internally, known as endonuclease. If they cut the nucleus externally, they are known as exonuclease. So the DNA polymerase have both the 3' as well as 5' exonuclease activity. This is an exception case because rest 2, that is DNA polymerase 2 and DNA polymerase 3 have only 3' to 5' exonuclease activity. Okay. And DNA polymerase 1 is responsible for DNA repair and gap filling, that is for filling the gap which will be formed while the nucleotides are formed. Okay, while the nucleotides are formed, to fill the gap, the DNA polymerase 1 is responsible, as well as DNA repair. DNA polymerase 2 is also responsible for DNA repair, whereas this polymerase, that is this enzyme, that is DNA polymerase 3, is main responsible for the replication. Okay, this is to be pointed down. Next, moving to the next enzyme, that is helicase enzyme. As I have already told you in this diagram, that the helicase enzyme binds to the one strand of the DNA and is responsible for opening of the strand. This is the function of the helicase enzyme. They separate the two strands. How will they separate the two strands? They break the hydrogen bond between them, and that's by the presence of sorry, by the hydrolase enzyme is present. By that enzyme, it is responsible for breaking the hydrogen bond. Okay. And it is also involved, this helicase enzyme is not only involved in the process of DNA replication, repair process and recombination process also it is involved. And in prokaryote, this helicase enzyme is called DNA-B, and in eukaryote it is called MCM27 helicase. This is very important because in exam it is mainly asked. Okay, what is the helicase enzyme of the prokaryote and what is a eukaryote? While reading this process separately, we will discuss in detail. Next, coming to the primase. What is primase? The primers are the oligonucleotide, means the chain of nucleotide that are complementary to the DNA template. So what is DNA template? So once the DNA stands open, one strand of the DNA acts as a template, means acts as a reference. So by seeing that template strand, another strand will be formed. That is known as the DNA template. So the primer gets attached to the DNA template, and what happens to how does this attach? Because they are complementary to each other. So these primer attach to the DNA template and forms a complementary bond with it. And next, what happens to the primer? The primer, what will happen? See, helicase enzyme will be attached. See, helicase enzyme will be attached to it, and the two strands will open. One strand will act as a template strand. This is known as the template strand. Okay. So both the sides, one strand will be act as a template strand, and this is the newly forming strand. This is the newly forming strand, lagging strand. Another one is known as the leading strand. What are they? We will discuss in the next subsequent videos. So what happens? This primer, this RNA primer, what will happen? They will attach to the template strand. They will form, here they will, this is the RNA primer. They will form the complementary basis to that of the DNA template. And then what happened? This DNA polymerase will bind to that of the RNA primer, and then the elongation or the formation of new nucleotide takes place. Okay. So to the primer, the DNA polymerase bind and elongate, and RNA primer provide free 3' OH substrate to bind with the DNA. So they provide free 3' OH end by which the DNA polymerase will bind to it, and the process of elongation takes place. Again, in case of prokaryotes, the DNA it is known as DNA G primase, and in eukaryote it is known as DNA poly alpha. Okay, alpha it is represent like this. Next, coming to the single stranded binding protein. As I have already told you, what is the function of single stranded binding protein? Is that when the two strands get open, this single stranded binding protein will hold the two strand in that position only. Otherwise, they will get as they both the strands are complementary to each other, they again will fold and get attached to each other, and the replication process can't take place. So in order to prevent from being wrapped up again. That's why the single stranded protein holds this two strands of the DNA and also prevents from the attack by the nucleus enzyme. These are the two main function of the single stranded protein. Okay, prevents the strand from hardening as well as prevents from broken down by the nucleus enzyme. As well as the prokaryotes, they are known as the SSB protein, and in eukaryote it is known as the RPA protein. Okay. Next, coming to the main enzyme, that is topoisomerase. What is the topoisomerase does? It mainly regulates the winding and the unwinding of the DNA strand. Suppose this is the DNA strand. Okay, this is superly coiled. Okay, supercoiling is done. So what does the DNA topoisomerase do? It mainly relaxes the DNA. Okay, it mainly relaxes the DNA and makes them tension free or stress free. Okay, so that smoothly the DNA replication can takes place. They are again of two type, that is topoisomerase 1, which how does they relax this strands? By relax this strand, either they make a single strand cut or both the strand they will make a cut and they will relax by twisting in the opposite direction. Okay, the single topoisomerase 1 forms the one strand break, and they are ATP independent and they are responsible for both the positive and negative supercoiling, mostly negative supercoiling, and topoisomerase 2 mainly forms the break on both the strand and they are ATP dependent and regulate the positive supercoiling. So this is the function of topoisomerase. Next, coming to the last part is the ligase enzyme. What is the form? It is joining. So main role of the ligase enzyme is joining, and ATP is required and it seals the gap. In case of eukaryotes, ATP is used as a form of energy, and in prokaryote, NAD plus is used as a form of energy. This is very important. So in mammalian case, there are two types of DNA. Ligase 1 is mainly responsible for joining the lagging strand, that is the Okazaki fragments joining. The DNA ligase 1 is responsible, and DNA ligase 2 is mainly responsible for the DNA repair process. How does they do? They form the covalent bond, phosphodiester bond between one nucleotide of the 3' OH of one nucleotide and 5' end of the another nucleotide. They join them. Okay, they form a covalent bond between three. There is a gap between the two nucleotide, one 3' end, another one 5' end. They will join that by forming a covalent bond. So this is the role of the ligase enzyme. Hope you have been clear what are the roles of the enzyme, what are the different role and what do they do in the replication process. This is all about the enzymes which are involved in the process of replication. Hope you have it clear. If you have any doubt regarding the enzyme, do ask in the comment section, as well as share maximum among your friends, because these are very basic questions which are mostly asked. Overally we read about replication, but each term we don't read. What is the role of each of them, which are mostly asked in the exams. In the next part of the video, we will be discussing about what is the replication in, how does the replication in prokaryote takes place, and in next upcoming video, we will be dealing with the replication in eukaryote. So this is all about the replication enzyme. Hope you share among your friends, and if you have not subscribed to my channel, do subscribe and have a nice day. Thank you.